Good morning everyone. Welcome to your biology class. This video is in continuity with the previous video of the chapter Human Health and Diseases. And the main objectives we are covering in this video are components of acquired immune system, types of T cells, response of B cells and the humoral immunity, response of T cells and the cell-mediated immunity, primary and secondary immune responses, and last is active and passive immunity. So first is components of the acquired immune system. So the acquired immune system has two components. First is humoral immune system. It is made up of various types of specific antibodies which are present in the blood, plasma and lymph. They defend the body against specific bacteria and viruses that enter the body fluids like blood and lymph. The term humoral is used for body fluids. B lymphocytes are responsible for the humoral immune system. It is also called antibody-mediated immunity. Next, cell-mediated immune system. So this system refers to the highly specialized cells of the immune system that work not only against pathogens but also against fungi, protistants that invade the host cells. These cells recognize the body's own cells and react against the transplants also. They also protect the body from their own cells if they become cancerous. The cell-mediated immune system is formed by the T cells. This is called cell-mediated immunity. Next, types of T cells in cell-mediated immunity. So cell-mediated immunity is carried by a subgroup of T cells called T cytotoxic cells. T cytotoxic cells have receptors on their surface that can recognize a specific antigen. On stimulation, activated T cytotoxic cells produce a clone of cells. Then the entire cell attacks the specific antigen or infected cell. The body contains specific T cells for every antigen. Activated T cytotoxic cells are specific to the target cell. They may kill the foreign antigen, the microorganism, but mostly attack the infected body cells and kill them by a variety of mechanisms. By destroying the infected body cell, host of the pathogen, they prevent the completion of life cycle of the pathogens. Now, cell-mediated immune response is used by the body to protect against pathogens like viruses by attacking virus-infected cells, certain fungi and protists that invade body cells. Tissue transplants, the T cells can recognize the body's own cells, that is, self against non-self. They attack and destroy the transplanted cells. And cancers, they kill the cells that become cancerous. So here are the types of T cells: naive T cell, regulatory T cell, memory T cell, T helper and T killer cells. Types of T cells. So first of all, helper T cells. They bind with the antigen presenting cells and detect infection, stimulate the production of both B cell and cytotoxic T cells. Next, inducer T cells. So it is not involved in the immediate response to infection and it mediates the maturation of T cells in thymus. Cytotoxic T cells, it detects and indirectly kill the infected body cells produced in response to a cell from helper T cells. And last is suppressor T cell. Suppress or reduce the activity of T cells and B cells after the infection had been controlled. Next, response of B cells and the humoral immunity. So when B cells in the lymph nodes come into contact with specific antigen, the receptors on their membrane recognize the antigen. The B cells are stimulated and divide repeatedly to give rise to a clone of plasma cells and memory cells. Clone is a population of cells derived from a single origin cell. Then All the plasma cells are genetically identical and hence they produce numerous identical antibodies which are released into the lymph, which eventually enter the blood. Most plasma cells can produce 2000 molecules of antibodies per second. They lived for a few days only. Memory cells produced, however, remain in the lymph nodes for 7 years and provide a rapid response during any second encounter with the same antigen. The antibodies either stick to the surface of the bacteria and speed up phagocytosis or neutralize the toxins produced by them. Millions of B cells, each being specific to a particular antigen, are present in the body at birth. Number of these B cells which react with the antigens of one's own body during embryonic development are rendered inactive and are killed, and this is the reason why antibodies are not formed to the individual's own antigens. Next is response of T cells and the cell-mediated immunity. So the cells also have receptors on their membrane that can recognize a specific antigen. On stimulation, they also produce a clone of T cells and produce specific antibodies, but the reaction is different. The T cells, instead of releasing the antibodies into the bloodstream, hold on to them and the entire cell attacks the specific antigen. The life of T cells is about 4 to 6 years. The body contains specific T cells for every antigen. Next, differences between B cells and T cells. So B cells they form a part of the antibody mediated or humoral immune system, and T cells, they form a part of the cell mediated immune system. B cells are processed in the bone marrow and hence are called B cells. T cells mature in the thymus gland and hence they are called T cells. B cells release the antibodies produced into the lymph which eventually enter the blood. T cells, the whole cell, directly attacks the antigen or infected cells. B cells, they defend the body against invading bacteria or viruses. They do not react against transplants or cancerous tissues. Whereas T cells, they defend the body against pathogens but also attack the transplants and the cancerous cells. Next, differences between two components of the immune system, that is humoral immune system and cell-mediated immune system. So, humoral immune system made up of B lymphocytes and cell-mediated immune system made up of T lymphocytes. It defends against the pathogens that invade the body fluids. It defends against the pathogens as well as fungi or participants that invade the host cells. Do not react against transplants and cancerous cells. It reacts against transplants and cancerous cells. Primary or secondary immune responses. When our body encounters a pathogen for the first time, the response is called primary response, which is of low intensity. And the subsequent encounter with the same pathogen elicits a highly intensified response called the secondary or an amnestic response. So, primary immune response. When a pathogen invades the body for the first time, there are very few B cells and T cells that have receptors which can recognize the pathogen. During the first encounter, very few lymphocytes are exposed to an antigen. The immune response is weak and this is called primary immune response. The exposure to an antigen triggers and two types of cells are formed, effector cells and memory cells. Effector cells produce antibodies that attack the infected cells to fight infections. They usually survive for a very few days, and memory cells do not produce antibodies or kill infected cells. They remain in the lymph nodes and get activated by a second exposure to the same antigen. They can remain in the body for decades. So, here, this picture, it is showing the processes in the primary immune response. So next, secondary immune response. So as a result, for the first infection, there is a large clone of memory cells, lymphocytes in the body that can recognize that pathogen or antigen. Second and subsequent exposure to the same antigen triggers a faster and a stronger response due to the presence of primary cells produced in the first encounter. This heightened immune response, which is more effective by subsequent exposure to an antigen, called secondary response. In the secondary response, there is a clone or effector cells and a clone of memory cells. The effector cells produce antibodies or attack the foreign antigen for the secondary response, and the memory cells remain in the body, capable of responding to future exposures to the antigens. So here, a picture which is showing a difference between a primary response and a secondary response. So, difference between primary and secondary immune responses. So, primary immune response refers to the immune response as a result of the first encounter of an organism with an antigen, whereas secondary response refers to the immune response as a result of the second and subsequent encounters with the same antigen. Primary immune response takes relatively longer for the immune system to respond, and secondary immune response, the response of the immune system is very rapid due to the presence of memory cells who get into action immediately. Primary immune response results in a feeble or mild response and declines rapidly, whereas secondary immune response results in a strong response that lasts much longer, maybe a lifetime. Individual may suffer from a disease, however, it results in the formation of memory cells that help in the secondary immune response. And in secondary immune response, individuals may not suffer from disease due to the heightened response. Next, active and passive immunity. So, active immunity, when a host is exposed to antigens, which may be in the form of living or dead microbes or other proteins, antibodies are produced in the host body. It is called active immunity. In active immunity, the body manufactures the antibodies against the antigen, and that is why such immunity lasts and protects the organism for a long time. Active immunity can occur in the body by various means. First, when the body is exposed to an antigen for the first time, it contracts the disease. The body not only produces antibodies but also produces large number of memory cells. When the same antigen strikes the second time, the memory cells produce a rapid immune response and the body does not contract a disease. It may last a lifetime. Active immunity is slow and takes time to give its full effective response. Alternatively, same active immunity can be obtained by injecting a vaccine that stimulates the immune system against the specific antigen. So, here is a picture acquiring immunity to infections, passive immunity and active immunity. Next is passive immunity. In this immunity, the organism's own body does not produce the antigens. Instead, ready-made antibodies from some other organism are introduced to protect the body against the disease. For protection against tetanus, diphtheria, rabies, the disease-causing bacteria or virus is introduced into another animal like horse or sheep. Antibodies soon appear in the horse or sheep's blood. From there, the antibodies are extracted and injected into humans to provide the passive immunity. Such an immunity is short-lived but can avert death. Mother's milk contains number of antibodies, immunoglobulins A, that protects your child from many diseases. The yellowish fluid, colostrum, secreted by mother during initial days of lactation has abundant antibodies to protect the infant. The fetus also receives some antibodies from their mother through the placenta during pregnancy.